Hi there. We're going to close our discussion with TCA cycle with a few slides on the regulation of the whole pathway. So this is the pathway again. And there are many forms of regulation. One, the common one is product inhibition. So what are the key products? As I said, it's electrons in the form of NADH, and NADH will go on allosterically and inhibit, okay, a variety of reactions. Actually, it's not allosteric. It's product inhibition. It goes to the active side if you have too much NADH and blocks the reaction. So they act as an inhibitor. Okay, the NADH molecules for all these reactions that generate NADH. Okay. So that's one way. Another product is succinyl-CoA. Okay, so succinyl-CoA will also act as a product inhibitor of this reaction here that generates it, but also it will inhibit citrate synthase, okay, as well. So product inhibition is one way, but there is another way which we have not seen up to now, but we'll see many more instances of this form of regulation. And this is a regulation of the enzyme by post-translational modification. What's the enzyme here? It's pyruvate dehydrogenase. Pyruvate dehydrogenase, which is the gateway reaction, the one that feeds acetyl-CoA to the TCA cycle reactions. And pyruvate dehydrogenase is active if it is not phosphorylated, but if there is a phosphoryl group on it, then it's completely inactive. Okay, who puts this phosphoryl group? It's actually pyruvate dehydrogenase kinase. So this is a protein kinase. We've seen sugar kinases. We've seen hexokinase, glucokinase, these kinds of reactions, but or PFK, for example, that put a phosphate on to sugars. In this case, this kinase is a protein kinase and phosphorylates a protein, pyruvate dehydrogenase. And when it does so, the substrate, so pyruvate dehydrogenase, now becomes phosphorylated, and now the substrate becomes inactive. Okay, this is a covalent modification. It's a post-translational modification, and in this case, the phosphorylation makes the enzyme inactive. In other cases, phosphorylation makes the enzymes active. Okay, so the outcome of phosphorylation, be phosphorylated or not, doesn't necessarily mean you're going to be always inactive. In this case, you are if you have pyruvate dehydrogenase, but on this site, it's a specific phosphorylation on a particular residue on the enzyme, a particular amino acid. Other phosphorylations have other outcomes. Now, how do you make pyruvate dehydrogenase again active? Well, you have to remove this phosphate. So there is a phosphatase specific, okay, that recognizes the phosphorylated form of pyruvate dehydrogenase and goes on and hydrolyzes this phosphate. And that converts pyruvate dehydrogenase back to its active form. Right. So here we have really two additional proteins, the kinase and the phosphatase of the target, that participate in the control, okay, of this enzymatic reaction. So in addition to that, we have all these other effectors that work on these regulatory proteins. Now, the kinase is activated by high amounts of NADH, which is of course a product of the TCA cycle. So if you have too much of that over NAD plus, okay, then perhaps doesn't make sense to go make more and more and more in the TCA cycle. So so then you activate the kinase, which inhibits pyruvate dehydrogenase, the gateway to the TCA cycle, and likewise with acetyl-CoA, if you have too much, then this enzyme, of course, produces acetyl-CoA. So if you have too much, it doesn't make sense to produce even more. So let's go and inactivate pyruvate dehydrogenase by activating its inhibitor, essentially. The phosphatase is regulated in a completely reciprocal manner, which obviously makes a lot of sense. So why do we do this? I mean, why don't we just have this compounds going directly and binding to pyruvate dehydrogenase and playing their role? Why do we need the middleman here, the middlemen and women here, enzymes, the pyruvate dehydrogenase kinase and the phosphatase? So the best way to see this is a higher order sort of bureaucratic control that really fine-tunes the activity of this enzyme. So the more layers of control you add, and here we have more layers of control, obviously. We don't just have the metabolites binding as inhibitors or activators of the enzymes. We have another layer with separate proteins, okay, controlling the activity, the output of the pyruvate dehydrogenase reaction. So we've added an extra layer, and that always allows for fine-tuning. And whenever you see this with these enzymes being regulated in such a manner, this is really an indication of a very important enzyme, which makes sense. It is the gateway reaction. And the cell goes into extraordinary lengths to add layers of control and truly fine-tune the output of that enzymatic reaction. It's a higher level government bureaucracy, so to speak. The more levels you have, the more control you have over the output. All right, so something about the evolution of the TCA cycle. It's important to recognize that TCA cycle is actually essential for life. Even if you only do fermentation, if you're yeast and you always do fermentation and you get your ATPs and some biosynthesis, you still need to do some of the reactions in the TCA cycle for biosynthesis. They generate extremely important intermediates for biosynthetic purposes. Succinyl-CoA, for example, is used for heme generation, and we've seen many of them are used for amino acids like oxaloacetate for aspartate, alpha-ketoglutarate for glutamate, and so on and so forth, and many others, and nucleotides as well. And if you go and mine the genomes of various organisms, and you go back, you find that some of them are actually missing some reactions, like the bottom one here, that takes succinyl-CoA and makes succinate. And this was at the time when we were discussing it. This reaction does not produce any obvious output of like the other reactions that's characteristic of the TCA cycle, such as electrons in the form of NDH or any intermediates that are unique and are used for in evolution and are used in biosynthesis, arguing that it was a late step. It was added late in evolution. And in fact, in some organisms, we have a modified TCA cycle. It's not even a cycle. It's more like a horseshoe kind of a pathway where the right side goes into the clockwise fashion, as we discussed, okay? And you generate intermediates and electrons, okay, in the form of NDH. And those electrons are fed into the reactions on the left side of the horseshoe, which go actually in the counterclockwise fashion, and they consume these electrons. So you generate on one side the electrons, you consume them on the other side, and this is actually very favorable thermodynamically. Remember, oxaloacetate it's an extremely, this particular reaction here from malate oxaloacetate is not very favorable at all, okay? So oxaloacetate going backwards. So in the canonical TCA cycle, oxaloacetate as a reason that's at very low abundance of all the intermediates. But if it goes in the other way, there is no problem, right? That's a favorable reaction. So this is sort of a self-sustained system where you get the electrons on one side and you feed them on the other side. So you are electron neutral and you get all your intermediates. What you don't get is feeding the electrons of NADH on some electron transport chain, charging a battery and making ATP. So if ATP is not your primary concern, and remember, with glycolysis, you're going to get some ATP. That's enough to get you going. Okay, it's enough for cancer cells and yeast, for example. And from this TCA cycle reactions, you get all the biosynthetic intermediates you need. So, if ATP, huge amounts of ATP is not your primary concern, you can be just fine doing all the biosynthesis you need in this scheme, the horseshoe scheme of the TCA cycle. And so that does make a lot of sense. It fits with the idea that this reaction was a closer, so to speak, close the circle so it can go around in our cells. Anyway, that's an idea anyway, but it's supported by experimental data actually. And with this, we close our videos and our discussion of the TCA cycle. So in the next sections, we're going to see what really happens to those NADHs, okay, that were produced in the canonical clockwise TCA cycle our cells do, for example. All right, so I will stop here and see you next time.